speaker is Bethany Moore from Ann Arbor, and the title is Periostin expression increases with age and regulates lung fibrosis.
All right, well, while they're putting that presentation up there for me, I'll just say that I actually want to thank Luca and the organizers for giving me the chance to talk. And I actually think I'm lucky to get to go last, because you'll see when I give my presentation that everyone who talked before me today gave a lot of context for what I'm about to tell you about as well. So we've been interested in a molecule called periostin. I think it probably doesn't need a lot of introduction in this audience. But periostin is a matricellular protein. And what that means is that it's a molecule that can be secreted and bind to the matrix, but then it also can interact with the cells that migrate over that matrix. And so in that way, it can transduce signals from the matrix to the cell to influence function. And this is a molecule that belongs to a family of molecules that include things like thrombosbondin, osteonectin, and things like that. And so the reason I think that the fibrosis community got particularly interested in periostin was this observation. I'm not sure, is this the top one, thanks, was this observation that periostin could then bind to fibronectin, which would localize periostin to provisional matrix, and then periostin could bind bone morphogenic protein 1, or BMP1, and in so doing could localize BMP1 to provisional matrix. That activated the ability of BMP1 to proteolytically activate lysyl oxidase, and in that way that promoted the cross-linking of monomers of collagen into collagen fibrils. And so it's been shown that if you overexpress periostin, you can increase the activation of lysyl oxidase and then increase stiffening, and we've just heard how stiffening might be important for the progression of fibrosis. Now, periostin can talk to the cells as well, and the way that it does that is by binding to cellular integrins, and there's been a whole variety of different integrins that have been shown to be able to bind to periostin. And the nice work from John Fahey's group had shown in the asthma models that if you overexpress periostin in bronchial epithelial cells, that then you could take the supernatant off of those overexpressing cells, put that on fibroblasts, and that could increase fibroblast activation, suggesting that this was a molecule that was promoting remodeling during asthma. So with that background, we were very interested in knowing whether there was any evidence in IPF for periostin to be upregulated and whether it could be contributing to the disease progression. And we were unfortunately scooped. But the first group to do this was Okamoto's group from Japan, and they showed that in the serum of control patients versus patients with IPF or fibrotic NSIP, that the serum levels of periostin that they could measure in these patients were in fact elevated over the serum levels that they could measure in control patients. And if you looked at the patients who had the highest levels of periostin at baseline and then asked what happened to their lung function 6 months later, you could see that there was an increase in the decline in DLCO in the patients who had the highest levels of periostin. Well, we were interested in this same question, and we first approached it through an immunohistochemistry approach. So shown here in DAB staining, which is brown, we looked at periostin expression in normal lung or in sections of fibrotic lung, and you can see that periostin stains very brightly the fibroblastic foci, so maybe some hyperplastic epithelial cells. We could see it in the subepithelial regions, as John Fahey had shown in the asthma models, and also in subendothelial areas. And these are fibroblasts that we grew from lung biopsies of patients who either had IPF or from normal lungs. And you can see that the message for periostin is about 3.5-fold increased in the lungs out of patients with IPF relative to the lungs of normal patients. We also did a similar study to the Japanese group where we had patients who we had a baseline blood sample on, and then we followed those patients over the next 48 weeks to ask whether they showed clinical progression as measured by a 10% change in FVC, a 15% change in DLCO, lung transplant or acute exacerbation or death, and then ask whether the levels of periostin at baseline predicted. And I won't spend a lot of time on this slide except just to say that you can see here in the non-progressors, the baseline levels of periostin were much lower than were the baseline levels of periostin in the patients who did progress. And so this is very reminiscent of what the Japanese group had published as well. Now, we were a little bit confused actually about where this periostin was coming from in the circulation, because we knew it could come from structural cells, but we weren't sure how it was getting to the circulation. And so we were interested in whether there could be a circulating cell type that was producing periostin. And so we had blood samples from patients, normal volunteers or IPF patients. These are just buffy coats stained with CD45, fixed, permeabilized, and stained intracellularly for periostin, and then gated on side versus forward scatter to try to look at the monocyte-sized population of cells. And you can see that the percentage of monocytes which express periostin is much higher in patients where we got blood from IPF patients as compared to normal controls. And because of the limitation of the amount of blood we had, we couldn't look at fibrocytes by flow, but we cultured the peripheral blood to allow the fibrocytes to be isolated in vitro, and then looked at the message for periostin between the fibrocytes from IPF patients versus normals. And you can see that in all but one IPF patient, we could detect message for periostin and essentially could see no periostin in the fibrocytes cultured from the normal patients. So again, suggesting that in IPF there may be a circulating population of cells that help produce periostin as well. So at this point in time, we were interested in trying to do some more functional studies, so we moved to the periostin knockout mice and asked the question of whether they were protected from fibrosis, and we got scooped again. And so the Japanese group had actually bred their periostin knockout mice to the BALB-C background, and this is their data showing that when they treated their mice with bleomycin, they could show that it was protected. Now, what was interesting is the mechanism by which they thought they were protected had to do with a lack of inflammation in response to bleomycin. So maybe we got lucky because we bred our knockout mice to the Black 6 background. We also show that they're protected. So here are the wild-type mice treated with bleomycin, developing an increase in hydroxyproline characteristic of fibrosis, and the periostin knockout mice treated with bleomycin are relatively protected. Now, because we were interested in that question of whether the circulating source or the structural cell source was important, we created bone marrow chimeras in every combination between the knockout and the wild-type mice. So here, when we transplant Black 6 bone marrow into a Black 6 mouse, this essentially recreates a wild-type mouse. Here, knockout into knockout kind of recreates the knockout mouse. And as expected, the wild-type mice develop fibrosis to bleomycin. The knockout mice are protected, but to be honest, we were really shocked to see that it did not matter whether you took the periostin production away from the structural cells or from the hematopoietic cells. In both cases, we protected the mice. So either this means that both sources are very important, or that there's just a minimal threshold that you have to be able to achieve for periostin to continue to promote fibrosis. All right, so at this point in time, we were quite convinced that periostin could regulate fibrosis, and now we were very interested in whether it could be therapeutically targeted. And it's very appropriate that I present this work in Italy because my collaborator on this work was Barbara Carnamola, who's from Genoa. And she had developed an antibody called OC20 in her melanoma work, which is actually able to block the interaction of periostin with the alpha V beta 3 and the alpha V beta 5 integrins. And she could show that that could prevent migration of melanoma cells using this particular antibody. And she was kind enough to share it for us. And the first experiment we did was a survival experiment. So we gave bleomycin to the mice on day 0, and we waited till after the inflammatory phase and started giving the OC20 or a control antibody on day 10 or day 15 after bleomycin. And you can see here that if the animals received bleomycin, and this was a high dose, with control antibody, they started to die during this 12 to 19 days after fibrosis or after induction of bleomycin. But if they were treated with OC20, they were significantly protected, and the histology was really impressively different. Well, we didn't want to measure fibrosis in these experiments where there was such a survivor effect, so we did a more traditional dose of bleomycin, which only gave us less than 10% mortality. But again, we gave bleomycin or saline on day 0, and then treated with OC20 or a control antibody on day 10 or 15, harvested the lungs at day 21, and you can see that whereas the control antibody-treated bleomycin mice developed fibrosis, there was a significant inhibition in the mice that were treated with OC20. So we're very excited about the fact that this therapeutic targeting of periostin seems to have some beneficial effect. All right, so because we were convinced that blocking periostin during the fibroproliferative phase was having an effect, we wanted to study the direct effects of periostin on lung fibroblasts. And so in this case, these are just mouse lung fibroblasts that we've treated with a dose-responsive periostin and looked for collagen messenger RNA. And you can see that in a dose-dependent fashion, we increase collagen. We also, in a dose-dependent fashion, can increase proliferation. And if we do a wound healing assay and measure the time it takes for fibroblasts to close a scratch wound, you can see that there's a dose-dependent increase in the ability of periostin to promote the closure of that wound, which is probably affecting both proliferation and migration. And in IPF lung fibroblasts, if we allow them to close the wound in serum-free media, it occurs much faster when there's periostin present in the media. But if we add Periostin and block with the OC20 antibody, we have an intermediate response. Now, also, as we've heard, we know that fibroblasts survive inappropriately during IPF, and so we wondered whether Periostin could be promoting a pro-survival phenotype in fibroblasts. So this is a mouse lung fibroblast treated for 90 minutes with Periostin, and you can see there's a dose-dependent, well, maybe not so dose-dependent, but there is an increase in XIAP, which is an inhibitor of apoptosis, and we have similar results for survivin and BCL2. So we also think that this molecule is stimulating a pro-survival effect. Now, as you've heard from Mauricio, both Anna and I were both interested in looking at aged mice with the gamma herpes virus, because this is a very nice model, because the old mice develop fibrosis to the virus, whereas young mice do not. And there are multiple reasons we've heard for why the aged lung might respond differently to environmental insults, but we wondered if Periostin was one of those reasons. And in fact, these are fibroblasts purified from old mice and fibroblasts purified from young mice. And you can see by ELISA that there's more Periostin in the old mice fibroblasts. And the fibroblasts from the old mice are protected from apoptosis, just like Victor showed this morning. And again, if we purify the fibroblasts from the aged infected mice, they have increased matrix production as compared to the fibroblasts from the young mice. And again, I've shown you that these are all effects that we know Periostin can promote. So we'd like to hypothesize that as the lung ages, this increase in Periostin may be one reason why the lung responds differently to these insults. So I'll just summarize by saying that the Periostin levels are increased in IPF patients. They do seem to show some correlation with disease progression. We're very excited about the possibility of this being a biomarker for progression that could be used in clinical trials. We know Periostin can promote fibrotic responses in mice, that the production of it by both hematopoietic as well as structural cells seems to be important. We're very excited about the fact that a therapeutic blockade seems to have some efficacy. We know that Periostin can promote many features in fibroblasts that seem to be pro-fibrotic, and that it increases with age and may contribute to age-related fibrosis. And so with that, I'll just thank the many people, but in particular my Italian collaborators, Barbara and Paola, for the antibody. I'll take your questions. It can come from a lot of places.
You had a tenfold difference between how much Periostin was coming from macrophages versus the blood, the pericytes or whatever. What are your thoughts about the major source in the lung in the context of fibrosis?
Right. Well, we have not done the experiments to directly measure the production by, say, epithelial cells versus fibroblasts versus monocytes, and I think that's an experiment that we can do in the mice, and so we will plan to do that. But I like the idea that the inflammatory cells might, through their chemokine receptors and chemotactic responses, get to the site of injury and prime the pump, if you will, by producing some Periostin that then may have effect. To be honest, the chimera experiments were a big shock to me. I thought for sure it was going to be the structural cells were the only important source. But now I'm not so sure. And so I think that that bears a lot of study.
So the significance of the fibrocyte production, which was low, is a bit uncertain at this
Well, and I
stage.
can't really say the fibrocyte data I showed you was message, not protein, and so I really can't address that protein level just yet. Hi, Rachel.
I really enjoyed your talk.
Thanks.
It's really curious, isn't it, that it can do so many things, and it's mainly binding integrins. Have you got any thoughts about which cell receptor might be important in terms of transducing subsequent proliferative and profibrotic effects?
Right. I mean, the only clue we have to that is that Barbara's antibody blocks alpha V, beta 3, and 5. Mm-hmm. And so it seems like if we block those two particular integrins, we seem to have some beneficial effect. Now, I don't know that that rules out other integrins or even potentially other non-integrin receptors that might be able to bind to Periostin. And so I'm hoping that maybe with some other antibodies that may block other regions of the molecule that we might be able to get some handle on that. But this is really pretty early work for us.
So follow-up question, if I may. If it's alpha V beta 5, do you think it's interfering with TGF beta signaling,
Right.
or promoting TGF-beta signaling.
Right, so I guess I should have said that there's clearly an autocrine loop with TGF-beta and periostin. So periostin will induce the production of TGF-beta, but TGF-beta also induces the production of periostin. And so I'm sure that, you know, if you can interfere with that forward feedback loop, that's probably beneficial. We know that some of the effects, if we block ALK5 inhibitors when we put periostin on the cells, then we can limit many of those effects. So I'm sure much of the profibrotic activity is through the induction of TGF-beta. But I will just say that the phenotype of the periostin knockout mice is quite different than the phenotype of TGF-beta knockout mice. And so I still think this is a better target for therapeusis, and that's particularly exciting to me.
Very nice, thank you.
Thanks.
In the meeting in Banff, there were about 30% of the papers and 50% of the posting talking about osteopontin. It went down of fashion progressively. There is a correlation between osteopontin and periostin.
They're both members of the matricellular family, and they both can influence cellular function. Certainly there's data for osteopontin to be able to regulate apoptosis. I am not as familiar with whether there's data about collagen production and proliferation. There's a huge literature on osteopontin that I'm not that familiar with, so there's probably that too.
Thank you.
Thank you.